The U.S. Centers for Disease Control and Prevention reported 5 cases of homosexual men's pneumonia in June 1981. The peculiarity of these pneumonia: a germ responsible for all of the cases is often not dangerous to humans. As time passed, an increasing number of incidents involving persons of diverse sexes and genders were documented. Affected individuals lost weight, developed fever and blisters, and had a compromised immune system. Scientists then attempted to understand what had occurred once the sickness proved lethal. Finally, in 1983, it became. It is undeniable that the human immunodeficiency virus, or HIV, is to blame for this illness. There are currently 2 treatments for HIV. But each day, more than 6,000 people contract the disease, many of them pass away. Scientists have been working unsuccessfully for the past 40 years to create an HIV vaccine, but this could soon change. The vaccination has a success rate of 97% in triggering immunological cells, which are important for warding off disease. HIV. So with that, I'll introduce myself as Clemens Steineck and let's speak about a potential HIV vaccine today. The virus that causes acquired immunodeficiency syndrome, also known as AIDS, is known as acquired immunodeficiency virus, or HIV. When we discuss 2 patients who had been cured of HIV, we have covered the topic on this channel. Therefore, the beginning will resemble one another quite a bit, but it is crucial to comprehend how the virus penetrates our cells. Sharing injection equipment or engaging in unprotected sexual activity are common ways that HIV is spread. Once in the bloodstream, HIV particles infect a subset of T cells. T cells are a unique class of immune cell that will be significant shortly. Once within a T cell, HIV begins to activate that cell's machinery and replicate its genetic material. HIV wants to splice its genetic material into the DNA of the host cell. When that occurs, the virus frequently goes dormant for a very long time. Furthermore, this is a horrible technique because our immune system is unable to detect what is wrong. But soon, the virus will turn itself back on, begin to replicate, and create fresh viral particles. Following their discharge, these particles are prepared to infect fresh T cells. Then, the entire process typically takes a few years, and throughout this time the infected person may not show any symptoms. They have no idea that they are HIV positive. However, AIDS might develop once enough T cells have been infected or killed. Weight loss, a fever, skin rashes, or white spots on the skin are all indications that you have AIDS. Lesions in the mouth area. Since the patient's immune system is no longer functioning effectively, other bacteria that ordinarily do no harm to us can suddenly begin to infect the patient. These infections are opportunistic, and they have the potential to cause the patient's death. There are numerous medications available today to keep HIV patients' bloodstreams free of active virus particles. Because of this, the condition can be somewhat controlled but not. Due to the fact that everyone in the globe may obtain these medications, it is imperative for researchers to develop an HIV vaccine. There are several reasons why it is particularly challenging to develop a vaccine against HIV, including the fact that nearly all vaccination studies have failed thus far. The typical operation of traditional vaccines is one of the primary causes. Typically, inactivated viruses or fragments of viruses are administered like traditional vaccinations. After receiving a vaccination, a person's immune system begins to identify the particles and fight them off. A crucial action is about to be taken. B cells, an immune cell subtype, begin to produce antibodies against surface proteins. Proteins known as antibodies are able to recognize particular structures known as antigens. Therefore, in this instance, antibodies identify the proteins on the virus's surface as antigens. Following vaccination, certain B cells continue to produce these antibodies, which circulate in the blood and give us immunity. And while most viruses are compatible with this, HIV is not. Since surface proteins are not identified, antibodies are frequently not generated in the first place. You see, HIV has a thick layer of sugar coating on its surface. There are so many sugars, in fact, that they reveal the underlying surface proteins. As a result, no antibodies are produced because our immune system does not interact with surface proteins. Therefore, we cannot develop immunity. Additionally, The fact that HIV is highly diverse is a second and crucial reason why there isn't an HIV vaccine. HIV has a comparatively high rate of mutation. Because there is a relatively high likelihood that the created particles are slightly different from the original virus when HIV starts to produce. Therefore, over time, HIV particles inside the same patient may become extremely different from one another. And amongst humans, the impact is considerably larger. Therefore, it is accurate to claim that each HIV case is unique and that there is no one form of HIV. Even if we had a traditional HIV vaccine, it would only fully protect us against some HIV particles. Therefore, the majority of HIV clinical studies have thus far failed for the reasons listed above. Only 2 vaccines in combination have been proved to reduce the risk of contracting HIV. The number of infections has decreased by 31% after this vaccination combination was developed in 2009. In the end, this type of defense also seems to disappear after a year or so. So that's it. However, HIV has one deadly defect that we may leverage to our advantage. HIV has a high imitation rate, which means that it alters its appearance to avoid being recognized by the immune system, as was already described. However, HIV cannot evolve everywhere without suffering repercussions. For the overall structure of the virus, some surface protein components are crucial. The whole particle may disintegrate dramatically if these areas are altered. Although HIV mutates very quickly, some sections are conserved, thus some components remain constant from patient to patient. And this is where the newly developed HIV vaccine is useful. In the past, supposed broadly neutralizing antibodies have been found in HIV patients increasingly frequently in recent years. The surface of harmfully neutralizing antibodies is recognized by conserved protein domains. Consequently, each of these antibodies was examined to determine exactly where they bind. His information allowed for the development of antigens, which are currently the main ingredient of this vaccine. However, there is a catch. These antibodies are extremely rare, which is why it took so long for their discovery. These antibodies can only be produced by one B cell out of a million. Therefore, it was necessary to adopt a novel method called germline targeting. We give our antigens to very particular B cells during germline targeting, and in this instance, we are discussing B cells, which have the capacity to produce broadly neutralizing antibodies. Since you might not. Okay, what are the initial findings? 48 healthy participants in a phase 1 clinical trial received the vaccination. No serious side effects were observed, and the vaccination seemed to have the expected result, therefore it seemed to be safe. In fact, 97 of the subjects had broadly neutralizing antibodies. Making these uncommon antibodies is remarkable, but we still don't know if this vaccination will fully protect us against HIV. So once more, this might become one of the major discoveries in vaccine research to date is this. I mean, you make me shudder. If you stop to think about it, that is truly absurd. I guess I got a little overexcited, but what do you think? Who are you? What do you think about this technique, and do you believe that HIV will eventually be eradicated? Please let me know as well. Please let me know what other subject I should explore next in the comments area. Okay, let's keep it brief once more. If you like this video, please subscribe if you're new here and click the bell to stay updated. I'll speak to you again later. You can watch this movie to learn how 2 people were cured of HIV. You might enjoy this video if you're curious in the AstraZeneca blood clotting controversy.